This is the continuation of musculoskeletal system. In this video, we will cover growth and development of bones, how bones grow in length and how bones grow in diameter. Bones consist of two types of tissue, bone tissue and hyaline cartilage. At birth, bone consists mainly of cartilage, which is firm but flexible. As the child grows, cartilage is replaced with bone tissue in all parts of the skeleton except the articular surfaces. The articular surface is the end of a bone that articulates or joins with another bone. Articular surface is covered in articular cartilage, which is a smooth, elastic layer of hyaline cartilage. Blood supply to bones is abundant because, like all living cells, bones and cartilage cells need a continuous supply of nutrients and oxygen and must excrete their waste. Long bones continue to lengthen potentially until adolescence through the addition of bone tissue to the epiphyseal plate. They also increase in width through appositional growth. Now we will see how bones grow in length. Growth in length of a bone occurs in the epiphyseal plates of hyaline cartilage at each end of the long bones in children and adolescents. In adults who have stopped growing, these ossify or become replaced by bone at the epiphyseal line. The epiphyseal plate is the area of growth in a long bone. It is a layer of hyaline cartilage where ossification occurs in immature bones. On the epiphyseal side of the epiphyseal plate, cartilage is formed. On the diaphyseal side, cartilage is ossified and the diaphysis grows in length. The epiphyseal plate is composed of 4 zones of cells and activity. The reserve zone is the region closest to the epiphyseal end of the plate and contains small chondrocytes within the matrix. These chondrocytes do not participate in bone growth but secure the epiphyseal plate to the osseous tissue of the epiphysis. The proliferative zone is the next layer toward the diaphysis and contains stacks of slightly larger chondrocytes. It makes new chondrocytes via mitosis to replace those that die at the diaphyseal end of the plate. Chondrocytes on the epiphyseal side of the epiphyseal plate divide. One cell remains undifferentiated near the epiphysis and one cell moves towards the diaphysis. Chondrocytes in the next layer, the zone of maturation and hypertrophy, are older and larger than those in the proliferative zone. The cells which are pushed from the epiphysis mature and are destroyed by calcification. This process replaces cartilage with bone on the diaphyseal side of the plate, resulting in lengthening of the bone. The more mature cells are situated closer to the diaphyseal end of the plate. The longitudinal growth of a bone is a result of a cellular division in the proliferative zone and the maturation of cells in the zone of maturation and hypertrophy. Most of the chondrocytes in the zone of a calcified matrix, the zone closest to the diaphysis, are dead because the matrix around them has calcified. Capillaries and osteoblasts from the diaphysis penetrates this zone, and the osteoblasts secrete bone tissue on the remaining calcified cartilage. Thus, the zone of calcified matrix connects the epiphyseal plate to the diaphysis. A bone grows in length when osseous tissue is added to the diaphysis. Bones continue to grow in length until early adulthood. The rate of growth is controlled by hormones, which will be discussed later. When the chondrocytes in the epiphyseal plate cease their proliferation and bone replaces the cartilage, longitudinal growth stops. All that remains of the epiphyseal plate is the epiphyseal line. Long bones stop growing at around the age of 18 in females and the age of 21 in male in a process called epiphyseal plate closure. During this process, cartilage cells stop dividing and all the cartilage is replaced by bone. The epiphyseal plate fades, leaving a structure called the epiphyseal line or epiphyseal remnant, and the epiphysis and diaphysis fuse. Now we will see thickening of long bones. How bones grow in diameter. Growth in width is due to the addition of cells on the inside of periosteum and is more active in children. While bones are increasing in length, they are also increasing in diameter. Growth in diameter can continue even after longitudinal growth ceases. This is called appositional growth. Appositional growth is a process of bone formation where new bone is added directly to the surface of existing bone. Osteoclasts resolve old bones that lines the medullary cavity. Osteoclasts on the inner surface break down bone, while osteoblasts via intramembranous ossification produce new bone tissue beneath the periosteum. Osteoblasts at the bone surface secrete bone matrix. The erosion of old bone along the medullary cavity and the deposition of new bone beneath the periosteum not only increase the diameter of the diaphysis but also increase the diameter of the medullary cavity. This process is called modeling. The osteoblasts differentiate into osteocytes. A balance between these two processes allows the bone to thicken without becoming too heavy. Although the length and shape of a bone does not normally change after ossification is complete, bone tissue is continually being remodeled and replaced when damaged. Osteoblasts continue to lay down osteoid and osteoclast reabsorbs it. The rate in different bones varies. For example, the distal part of the femur is replaced gradually over a period of 5 to 6 months. Development of bone tissue begins before birth and is not complete until about the age of 21. Long, short, and irregular bones develop from rods of cartilage or cartilage models. Flat bones develop from membrane models and a sesamoid bone develops from tendon models. Bone development consists of two processes. First one is secretion by osteoblast of osteoid, that is collagen fibers in a mucopolysaccharide matrix which gradually replaces the original cartilage and membrane models. Next process is calcification of osteoid immediately after its deposition. There are two types of arrangement of collagen in osteoid. First one is woven or non-lamellar bone. Collagen fibers are deposited in irregular bundles, then ossified. This primitive bone structure is a part of a normal fetal development occurring during ossification of bones that originate as membrane models, for example, skull bones. In adults, it is also present in bone tumors and healing fractures. Next is lamellar bone. The collagen fibers are deposited as in woven bone, organized into characteristic lamellae found in combat and cancellous bone, then ossified. This occurs when cartilage models are replaced by bone and in healing of fractures. In long bones, the focal points from which ossification begins are small areas of osteogenic cells or center of ossification in the cartilage model. This is accompanied by development of a bone collar at about 8 weeks of gestation. Later, the blood supply develops and bone tissue replaces cartilage as osteoblasts secrete osteoid components in the shaft. The bone lengthens as ossification continues and spreads to the epiphysis. Around birth, secondary ossification center develop in the epiphysis and the medullary canal forms when osteoclasts break down the central bone tissue in the middle of the shaft. After birth, the bone grows in length by ossification of the diaphyseal surface of the epiphyseal cartilage, and growth is complete when cartilage becomes completely ossified. The exact mechanisms by which bone development is triggered remains unclear, but growth factors and cytokines appear to play a role.